Here we go, DNA and protein synthesis. DNA. Oh, we don't like the cookies in this one, so now we're opening the new one? Okay. I like
There's
this.
no chocolates in there.
Oh, there's no chocolate chips? Oh, you guys are picky. I almost bought extra chocolate chip cookies for you. And I thought, oh no, there'll be people who like oatmeal and white chocolate chip.
White chocolate chip's delicious.
Oh, okay. Shh. All right. It has raisins. Oh, what have you got against old grapes?
They're disposable.
I don't like it.
Oh, sad, 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 sad. All right, DNA and protein synthesis. DNA and protein synthesis. All right, DNA, locations in the cell, function, structural differences. DNA versus RNA. Make sure, any questions on DNA versus RNA? Because I don't want to spend a lot of time on it. This is something, you've learned about multiple times. Know what kind of sugar, deoxyribose versus ribose. Double-stranded versus single-stranded. Can't leave the nucleus, DNA can't leave the nucleus. RNA can, and this is why the central dogma of biology, the central concept we see over and over and over again, the driving force of evolution, DNA is used to make messenger RNA, which is used to code for proteins. And proteins make up traits, or give us our traits, our characteristics, okay? So I suggest that, I know we had a T-chart in our notebooks over DNA versus RNA, and the characteristics, go back and review that T-chart in your notebook. 3 parts of a nucleotide. The nitrogenous base is bonded to the sugar, and the sugar is bonded to the phosphate. The structure of DNA, and so now I got to go and give myself a little whiteboard to write on. Yeah, so the backbone of I heard no. The backbone of DNA is sugar, phosphate, sugar, phosphate, sugar, phosphate. And of course, if it's DNA, it's deoxyribose sugar. If it's RNA, it's ribose sugar. And then coming off of the sugar, you have your nitrogenous base, adenine, guanine, cytosine, or thymine. So this is your nucleotide. And again, the intent of this review is it's not possible for me to really give you the entire semester over again, but I'm doing my best. All right, so ATCCCGGT. All right, ATCCCGGT. We're going to write that down. ATCCCGGT. So if this is my strand of DNA, what process is it to make the other side of the DNA? It's replication. So if I've got an A on the one side, what do I put on the other side? T, A, G, 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 C, C, A. Are we all totally comfortable with that, right? Okay. I would like you to review the 2 different categories of nucleotides. Okay. You need to know that A bonds with T and G bonds with C. Okay. That's only in DNA. So, when we take, let's say that we take, and you have to be very, very careful when you're transcribing. Okay, so the process that we just did was replication. Right, where you take one strand of DNA and well, actually I didn't even replicate. Let's scratch that. That's not replication. But I was showing you what a double-stranded DNA molecule would look like. The process of replication is where you have your DNA, it uncoils, and then it unzips. And free nucleotides that are around, and I'm drawing these little T-shaped things, are the free nucleotides. They are going to be coming in and bonding at the complementary base pairs. So we have these nucleotides nitrogenous bases hanging off. We call this the replication fork. And the enzyme, DNA polymerase, that is going to facilitate the adding on of nucleotides can only work in one direction. So on one strand, it works from the end this direction. That's the leading strand, 5' to 3'. And then, so this end is the 5' to 3', and this end is the 5' to 3'. And because DNA polymerase can only attach in one and run in one direction, you have the leading strand, and then you have DNA polymerase there going in this direction, and that's called the lagging strand. So this one is going to work and replicate in this direction, and then let go and reattach farther up and replicate a chunk and then go. And then those little separate segments get stuck together. And the end result of replication is you end up with 2 new strands of DNA, or 2 duplicate strands of DNA. Half of it is old and half of it is new. So that's why we call it semi-conservative. And to replicate means to copy. Okay, so leading strand and lagging strand. All right, I'm gonna go away from this. So it's gonna go away. Don't panic. If you miss something, you can always watch it again or stop me. Let's see, similarities. No, the similarities and differences between DNA replication and protein synthesis. Okay, we'll get to that. We just talked about replication involves DNA polymerase, okay, and we're forming another strand of DNA. Transcription involves DNA as your template, RNA polymerase because you're making a strand of messenger RNA, okay. So what does that look like? So I like to go left to right. A, T, C, G, C, A. Okay. So this is my strand of DNA. RNA polymerase is going to be the enzyme that is going to be responsible for reading the bases and forming what? What is formed from transcription? What kind of RNA? Messenger RNA. Okay? So we're making messenger RNA using RNA polymerase. And the base pairing rules are a little bit different when we're making RNA, because if we have an A on the DNA, what are we going to have on the RNA? A uracil. Okay? And if we have a thymine there, what are we going to get? An adenine, and then guanine, cytosine, guanine, and uracil. So that is transcription. Translation. Now we have to go to a new language. We're going away from the language of nucleotides, and we're getting into a language of amino acids. So that's why we're translating. We're changing languages. In translation, first of all, where did this process occur that we just talked about? It's in the nucleus. So the messenger RNA leaves the nucleus, comes out into, and here's our strand of messenger RNA. U, A, G, C, G, U, A. I just added an A on the end. So, what's the next step?
tRNA.
tRNA yet? What do you need before the tRNA? Ribosomal RNA. The ribosomal RNA is going to come and it's going to set, and it's 2 subunits. and it attaches. Why did I just cover up 3 bases? Because it reads one codon at a time. Okay? Okay, one codon at a time. And so this is the rRNA. It's ribosomal RNA. Its job is to read the messenger RNA. This is messenger RNA again. And now this is when we talk about that other one, the transfer RNA, which comes in and it looks kind of like this. And it has 3 bases on the end and a specific amino acid on the other end. So if your bases on your messenger RNA are UAG, what is going to be the complement to UAG? AUC. So this is called the anticodon. The anticodon. And I happen to know from memory, because I always start out with these same 3 bases, that this codes for methionine. And so this is where you would go and you would take that chart, the circular chart, or the rectangular chart, and you work from, on the circle, you work from the inside out. Do you remember that? Yeah. Okay, and you can find that in your textbook. If it's the box chart, then you just start with the left, first base in the code, top. Does anybody need to see that again? To come up with the amino acids from the chart, from the messenger RNA?
What's it called? For making the amino acid, the coding would be the mRNA?
Okay, most of the charts that you're given give you messenger RNA codons. Okay? So you have to look at what you're being asked for. Okay? We did a lab where we used the transfer RNA, but it said use the transfer RNA. So you have to be careful readers and know what are you supposed to use to come up with your amino acids. The messenger RNA or the transfer RNA. Okay, you guys, this is very sensitive and it's picking up all of your conversations. Yeah.
So on the final, when we're using like codons and like trying to code like proteins and stuff, do we have to memorize like what each thing codes for, or we can have?
You do not have to memorize the codon chart. You're going to be always given that. Okay? Just like on the test. All right? So we've already talked about frame shift and deletion mutations and how those things impact if you have an insertion or a deletion and how that is a frame shift. And then you would be able to read the amino acid codon chart that was used in the chapter and complete it all. All right? So I'm going to stop here and upload this video, and you guys can take a breath.